Dear friends, greetings. Today we can discuss about genetic code. What is genetic code? Genetic code is a dictionary that corresponds with sequence of nucleotides and sequence of amino acids. Genetic code is a set of rules by which information encoded in genetic material, either DNA or RNA sequence, is translated into protein by living cells. The term genetic code given by Gregor Gamma. Next, move on to discovery of genetic code. The first two scientists are James Watson and Francis Crick. To understand how proteins are encoded begins after the structure of DNA was discovered by James Watson and also Francis Crick. Both the scientists are next. Greg Gamow postulated that a three-letter code must be employed to encode the 20 standard amino acids used by living cells, which are build up the proteins. Those amino acids alone used for synthesis of proteins. Next, introduction about genetic code. The letters A, G, T and C. A means adenine, guanine, thymine and cytosine, which corresponds to the nucleotides found in DNA. They are organized into codon. These nucleotides, three nucleotides join together to form the codon. The collection of codon is called genetic codes. For 20 amino acids, there should be 20 codons. Each codon consists of 3 nucleotides. Each codon should have 3 nucleotides to impart specificity to each of amino acids for a specific codon. From this, one nucleotide, 4 combinations. For example, 2 nucleotide means totally 16 combinations. 3 nucleotide means 64 combinations. Most suited for 20 amino acids for genetic code. This picture shows from DNA to which codes the protein synthesis. For example, double helical structure of DNA, which consists of nitrogenous bases plus phosphoric acid and also ribose sugar to form the nucleotides. The three nitrogenous base codes for one amino acids. Next one, genetic code can be expressed in a simple table with 64 entries. Here 4 nitrogenous bases, for example UCAG, this is for first place as like first position, second position also UCAG, then third position UCAG. In this way, these nitrogenous bases alternately we can arrange it. For example, first position U common one, next for second position U also common, third position UCAG. These 4 nucleotides alone we are alternatively we can written as In this way we can returning means we can get that 64 codons. Next, codon and its type. Genetic code is a dictionary consists of genetic words called as codon. Each codon consists of 3 bases. Triplet codon are called as codon. There are totally 64 codons there. Among the 64 codons, 61 codes for 20 amino acids found in protein. Those 61 only for used as a protein synthesis amino acids, 20 amino acids. Remaining 3 codons do not code for any amino acids. These only codes for stop codon. Next one, type of codon. Sense codon. Second one is signal codon. The signal codons are which gives the signal either starting of protein synthesis or otherwise stop of protein synthesis coding. Next, the sense codon means the codon that code for amino acids are called as sense codon. Any codons which codes for amino acid means that is called as sense codon. Another one is signal codon. Signal, those codons that code for signal during the protein synthesis or translation process are called as signal codon. For example, AUG is a initiation codon. Initiation means which starts the protein synthesis. Another UAA, UAG and UGA. These 3 codons which are stop the protein synthesis process, so called as stop codon, which give the signal for stop the protein synthesis. There are 2 types of signal codon, termination codon and initiation codon. Termination means already we know stop codon. Initiation means start codon. Termination codons. UAA, UAG, UGA are termination codon or nonsense codon and are often referred to as amber, Aker and apple codons, opal codons. These are the terminating one. Next one is initiation codon. AUG is the initiation codon. It codes for the first amino acid in all proteins. AUG means which indicates the methionine amino acids. All the starting point, it codes for methionine in the eukaryotes and formyl methionine in prokaryotes. Methionine especially initiate the protein synthesis, especially for eukaryotes as well as prokaryotes. Next one, anticodon. The base sequence of tRNA which pairs with codon of mRNA during the translation is called as anticodon. Generally, mRNA, the 5' to 3' direction, nucleotides arranged it. Next, the tRNA, which consists of 3' to 5' direction, also nucleotide basis. This is called as anticodon, complementary to each other, codon as well as anticodon. For example, A means U, G means C, G means C. Here first anticodon A, codon U, second one G as a anticodon, C as a codon, as like third one G as a anticodon, C as a codon, normal one. But during the oval position, the instead of C will be translated to some other nucleotides. Difference between codon and anticodon. Codon could be present in both DNA and RNA, but anticodon is always present in RNA, never in DNA. Codons are written in the 5' to 3' direction, where anticodons are usually written as 3' to 5' direction. Anticodon of some tRNA molecules have to pair with more than one codons. Codons are sequentially arranged in nucleic acid strand, while anticodons are discretely present in cells with amino acid attached or not. Codons defines which anticodon should come next with amino acid to create the protein. Here mRNA and also tRNA, each one complementarily one by one amino acids will be added. In this way, protein synthesis will be taking place. Anticodon helps in bringing a particular amino acid at its proper position during the translation process. Codon and also anticodon nucleotide bases are complementary to each other. Then only the protein synthesis will be sequentially going on. Dear viewers, thank you for your support. Thank you.